So in biochemistry today I'll try to cover with amino acid, proteins and carbohydrates, not with the metabolism part, about the structure and the basic information part of the same. Okay, if we have the time, we'll go ahead with a little more. Let's see how we go about today. Okay, again, as we are dealing with a basic subject, in the class we'll talk about having the concepts clear, and with the MCQs we'll add on the tidbits bits needed, to solve it. So we'll be talking about very basics today. So let me start with amino acids maybe. Okay, so we all very well know what these amino acids are, right? They are nothing but the monomer units which we require for the formation of proteins. So all our amino acids, they just join together and they form the chains of proteins. Now one very important point which we have to notice over here is proteins contain only L-alpha amino acid when we talk about animal protein. What is L-alpha amino acid? I'll be just talking in a while. And before I talk about L-alpha amino acid, let me just talk about how the structure of an amino acid is. As we all know, amino acid, if you notice, we have a group here that is NH2. Do you notice this group over here? This is an amine group, NH2. And if you notice here, that is COOH. Okay, this group is your carboxylic acid group. So we have an amine group and an acid group, both in amino acids. Now very tricky, amino acids, they are both base and acid at the same time. Okay, yeah. Now if you notice, we have a carbon here to which we have one hydrogen, and on the other side we have this R. What is this R group? R group is nothing but the side chain. It could be any side chain. Okay, so this is the basic structure of amino acid. Okay, when we say this alpha carbon, this carbon which is just next to the carboxylic acid carbon, it's called as alpha carbon. Now whenever we have this R group attached to the alpha carbon, such kind of amino acid wherein the side chains are attached to the alpha carbon, they are called as alpha amino acids. Are you all clear with this now? What is alpha amino acids? Now let's talk about what is L and what is D. Okay, we have been talking about L and D as dextro and levo, if you remember. That's what is a general thing we usually have been talking about, right? But what has happened now? This L and D terminology was introduced because of the rotation of light when it was put into certain mixture. Sometimes the polarized light we are talking about. Polarized light is a light which has a direction. Okay, this polarized light, when it would rotate to the right, it was called as dextro-rotatory. When it would rotate to the left, it was called as levo-rotatory. Okay, that is how we got these terms as D and L. Okay, later when they studied, they found that certain structures, certain type of structures, they rotate the light to D side, the dextro side, and certain type of structures, they rotate the light to the left side. So based on these, the structures were called as D and L. But unfortunately what happened is, not all structures fit exactly into the criteria of D and L. Okay, I don't want to confuse you. What I'm telling right now is D and L was defined on the basis of the polarization of light. Later that definition was applied to the structure, right? But that a particular structure, for example, the side chain is on the left side or right side, based on that it would rotate the light to left or right. But it wasn't totally applicable. But still D and L is used for classifying the structure, defining the isomers, wherein The side chain and H bond are flipped around. So let me just draw you. If you want to have a D and L structure of this molecule, now what is this molecule? We have C, we have H, we have R group, and we have COOH, and we have NH2, right? On this side, I'm sorry, there was no space, so my diagram is a bit bad over there. If you want to rotate it around, what we can do is we can have these R and H exchanged. Okay, now when we talk about this rotation, this is just not a simple, rotating of the molecule. It's a mirror image, but this mirror image formation requires breakage of the bond. This mirror image gives the D form and the L form. Okay, so this is the D and L isomers of a particular amino acids. Fine, but now when we talk about the alpha is nothing but wherever your side chain is attached to a carbon atom that is just next to the carboxylic group. The first carbon atom after the carboxylic group, to which whenever the side chain is attached, it would be called as alpha. So just separate out the term alpha and L and D. Okay, now since in mammalian proteins, most of the mammalian proteins, we have L-alpha amino acids, we'll not be much talking about the other types, the beta and other things. When we go ahead with our discussion further, you will notice that we are all the time talking about the alpha amino acids. Okay, shall we go ahead with basic information? All clear with that? Okay, now when we talk about amino acid, you all very well know that amino acids are the basic building blocks of protein, and these amino acids are coded in our DNA. These amino acids, where are they formed? They are formed in an organelle which are called as ribosomes. Others, are you able to see the slides? What I'm talking about is, we have, where do these amino acids they form? They form in ribosomes, if you remember. Hopefully you're clear with that when I say that, right? And from where the information comes? Information comes from nucleus, from the DNA. Okay, DNA has these information stored as codons. So we have a triplet, that codes for a single amino acid. Okay, so yeah, this is a triplet codon that codes for one amino acids. Now what happens is, when we'll be talking about these codons and other things when we go to the details in the nucleic acid again, but right now what you need to know that human DNA, it codes for 20 amino acids, and these 20 amino acids, they are the one that acts as building blocks of protein. So we have, and where the proteins are formed are in ribosomes. So information comes from DNA to the ribosomes, and these ribosomes later make the proteins. Now the L-alpha amino acids present in proteins, we have various types, based on their structure. We have various classifications for the amino acids. In that, the main one, we have these the amino acids with aliphatic side chain. Most of the questions of amino acids are concentrated across two charts. One is about this structural chart, and the second is about the essential and non-essential amino acids. Okay, so although I'll be talking more about the other parts of amino acids, your role plays more important in remembering these charts when you talk about MCQ perspective. Is that okay? If the video gets too bright, just remind me, because I'll not notice it. As soon as the sun becomes brighter, it will be too bright. So just remind me, okay? So okay, let's talk about the amino acids with aliphatic side chain. In this we have this glycine, alanine, valine, leucine and isoleucine. Please don't postpone it. Have a first read now and here itself. Glycine, alanine, valine, leucine and isoleucine, these are aliphatic side chain. Then we have the one containing the hydroxylic groups. In that we have serine, threonine and tyrosine. the one containing sulfur atoms. This is a very important one. This is very commonly asked in MCQ. In that we have cysteine and methionine. Cysteine and methionine are the sulfur containing amino acids. The one which have the acidic group, this is a simple one. In that we have aspartic acid, asparagine, glutamic acid, glutamine. Actually, cysteine, Hemlata, is a derivative of cysteine. It is a post-translational amino acid. Right now we are talking about the basic amino acid. That is why that one is missing. Cysteine is actually derived from cysteine. So it has to be sulfur containing. So once you know that cysteine is derived from cysteine, you will automatically put that into sulfur containing. So we'll come across that slide wherein we come to know from where the cysteine comes. Okay? Hopefully I'm not confusing you. Right now you need to remember that cysteine and methionine are the very important sulfur containing amino acids. Right? Then we have this amino acid that contain the basic group. In that we have arginine, lysine and histidine. And then we have the ones containing aromatic ring, that is histidine, phenylalanine, tyrosine, tryptophan. Now in this chart you will find some of the groups that fit into two categories because they have the two types of structure. That is why we have the number going more than 20 over here. Hopefully that doesn't confuse you. It's a chart and this is a very important chart. You need to remember it. You'll definitely have a question from this chart in the upcoming. Then we have one that is amino acid. We'll be talking about this amino acid later. That is proline. Now we just now talked about the structure of the amino acid. All the amino acid, they have these central carbon attached to a carboxylic group and an amino group and an hydrogen atom. But proline is an exception. Now let's just see the structure of the proline. Okay. Now in the basic structure we saw that there is COOH and NH2. When we talk about the structure of the proline, let's locate where COOH is. Here is our COOH. Do you notice that? And where is NH? We have NH here. But NH2, what has happened to one more H of NH is, this is the H hydrogen and this is your alpha carbon. This was supposed to be your side chain. But if you notice, what happens is this side chain gets attached to one of the amine group. Because of this, it forms a kind of cyclic structure. It is not straight. Rather than being straight, it goes and interacts with the hydrogen of the amine group and forms a cyclic structure. That is why proline is not an amino acid. It forms a cyclic structure. That is why it is called as amino acid. Okay, that's an MCQ over there. Let's go ahead now. Now there's one more thing to notice. I'll just tell you one term that is called as chiral. Yeah, chiral. Thank you, Kirti. Chiral. I was just planning to write it on the board. So we have this term chiral. Term chiral, chirality, is nothing but a synonym for asymmetric. The term chiral means asymmetry. Now we'll see how it acts. You have COOH here, right? You have NH2 here. You have an hydrogen here and you have an side chain. If you notice this carbon, it is attached to 4 different molecules, right? Such carbon atoms which are attached to 4 different molecules, they are called as chiral carbons. Such carbons which are attached to 4 different molecules, they are called as chiral carbons or asymmetric carbons. And then they just notice these 4 things which are attached to this carbon. Do you notice the carbon over here? Do you notice this carbon? Do you see what it is attached to? It is attached to, on one side it has carboxylic acid, on the one side it has NH2, on the third side it has hydrogen, and the fourth side it has this side chain. All 4 different things are attached to it. Since all 4 different things are attached to it, it is asymmetric. Right? Such asymmetric are called as chiral. Now, because they are chiral, we can have isomers. Since they are chiral, we can have them rotated and we can have these L and D isomers. Are you getting? Now imagine a scenario if I remove this side chain, this R, and put H here. Let me remove the side chain and replace it by H. If my side chain is H, does our alpha carbon remains chiral now anymore? No, it will become symmetric because it has 2. It doesn't remain asymmetric anymore. And this is the scenario of glycine. Okay, so there is an MCQ here. The alpha carbon atom of all amino acid except glycine is asymmetrical. Fine? Such asymmetric centers, if we had asymmetric centers, we could have stereoisomers, that is enantiomers. Enantiomers are nothing but the mirror images we were talking about, that is nothing but the DLN amino acids. But if we don't have an asymmetric chiral atom, we cannot have D and L amino acids. I mean, we wouldn't have the enantiomers or the stereoisomers. Hopefully you know the term enantiomers or stereoisomers now. You understand the term. This term is used for the mirror images, that is D and L isomers. There are few things which you can grasp from this slide, and I don't know how, but such information will definitely help you in solving certain MCQs and orienting your brain there when the question comes. Okay, now one very important point again is coming over here, which we have noticed in the first slide. Only L amino acids are incorporated in mammalian proteins. Don't ever forget that. Okay, now if you notice, glycine is the one that has, instead of any other side chain, it has hydrogen. If you remember, hydrogen could be the smallest side chain possible, right? No, enantiomers are present for all the other. I'm sorry, I'm just shifting to Soumya's question. Soumya says enantiomers are only for glycine. No, it's just the other way around. Enantiomers are only for all the other amino acid except for glycine. Glycine cannot have enantiomers because it is symmetrical, right? Even if you just cut down its H, a side chain and H, and it rotate it around, it will be the same. It cannot have 2 forms. Right? Hope all of you got it. Okay, I'll shift back to the thing which we were discussing. We were discussing about the L amino acid are the only ones which are incorporated in protein. Did I say anything else there? That's it, right? Yeah, I was talking about glycine having the smallest side chain. If you remember, glycine has a side chain as H. It is the one that has the smallest side chain. So obviously the glycine is the smallest amino acid available in the animal amino acids. Okay, that makes one more MCQ. Keep a note of it. Make a habit of picking up the MCQs, guys. Okay, now let's talk about the ion properties of the amino acids. We have been discussing that amino acids have both acid and the basic group. This is the acid group and this is the basic group. Okay, since it has both acid and amine group, now it becomes tricky. Its pH, its isoelectric point, all these things becomes tricky. We'll see how it becomes. So it has both acid and it is both acid and a base at the same time. At a certain pH known as isoelectric pH, amino acid, it exists as an ionic form having no net charge. Please understand my statement at this moment. I'll explain you in the diagram in the next slide. Okay, isoelectric point is a pH at which amino acid exists as an ion without having a net charge. Its net charge is neutral. Okay, how it happens? Let's go back to the diagram and probably then we'll come back to this again. Okay, let me just complete this so that we don't confuse you. Okay, this is the side chain. This was your NH2, and this side we had COOH. Okay, that was the basic structure. What has happened is this COOH, every acid has a tendency to lose H plus. Hopefully that's a basic. You remember that every acid has a tendency to lose H plus. Similarly, COOH has that tendency, so it loses H plus. It goes to the base. Every base has a tendency to take up H plus. So this base takes up the H plus and it becomes NH3. When it takes up one H plus, it will receive a positive charge. Right? When COOH loses one H plus, it loses one H plus, so it receives a negative charge. Okay? So there is deprotonation. Deprotonation, you understand what is deprotonation? Deprotonation is losing of a proton. Proton is nothing but a positive charge and it's H plus. Okay? So the deprotonation is happening on this side and protonation is happening on this side. And you know, the great thing is all this is happening in the same molecule. Most of the time we talk about this happening from one molecule to another, but when we talk about the amino acids, it's happening in the same molecule. Right? But the thing is that, you know, at a particular pH, this happens, and a total at an isoelectric pH, what happens if you calculate the negatives and the positives, they'll be all same, and having, the total thing is neutral. If you just calculate, yeah, it's zero charge. Net charge is zero. So that is a zwitterion. That is called as zwitterion, and that pH is called as isoelectric point. Okay, at a certain pH known as isoelectric point, an amino acid has no overall charge, since the number of protonated amino groups, that is the positive charge. You remember now what was protonated amino group? And the deprotonated carboxylate group, that is the negative charge, are equal. The ions produced at the isoelectric point have both positive and negative charges, and these ions are known as zwitterion. Right? Okay? So that ion what is formed, right, which has both positive as well as the negative charge, it is called as zwitterion, and the pH at which it exists, it called as isoelectric pH. Clear with that? Or clear with that? Yeah, zwitterion as such is neutral because the charges, they balance each other. And you can say zwitterion is neutral. Zwitterion has both positive charge and a negative charge. Zwitterion exists at a particular pH, that is called as isoelectric pH, and any of these statements could be used as framing of MCQs. And they are. Clear? Right. Let's come back to Henderson-Hasselbalch's equation. Okay, when we will be talking about Henderson-Hasselbalch's equation, this equation is gonna be useful to you in answering one or two of the questions in pharmacology as well. Okay, so be open to this, and I'll just try to explain it whatever is needed for today's portion. What it expresses, Henderson-Hasselbalch equation, it describes the derivation of a pH as a measure of acidity. Every acid has a property to dissociate. Right? Every acid gives a proton. Just now we saw, right? So every acid has a property to dissociate in a proton and a remaining part. We'll talk what remaining part is. So when a proton separates, we get a proton and there is some remaining part. Fine. So with this ability, this capability, we define the pH as a measure of acidity. Let's go to the equation, and then we'll come back to this slide, so probably you will be able to understand it better. Okay. So when we talk about any acid, this is an acid. Okay. H is nothing but a hydrogen. Every acid has an hydrogen. A is nothing but the remaining part of the acid. For example, if I say HCl, in that your H is this H and A is nothing but Cl. That's what I'm trying to say when I write HA. Okay. When I write H2SO4, H means one of the H and A means HSO4. Do you understand that now? What does H and A mean? Great. Now whenever I dissociate, what happens? We have this H plus coming out, that is called as deprotonation, and we have this A minus because H plus is lost, so remaining part will have a negative sign. Got the sun? 1 second. Okay, I'll just is the video okay? I mean, tolerable or too bright? Too bright. 1 second. Yeah. Now, you understand this statement now? HA represents the acid and A minus is the conjugate base and H plus is the proton. Hopefully after this you will be able to understand what your keynotes say. Okay, I want you all to read the keynotes immediately after today's class, especially about these equations. This information we go ahead. Hi Jyoti, good evening. The Henderson, you know what we have is, for example, if we had HA and we dissociated it, what we get is H plus and A minus. If we take the ratio of the dissociated part, not take a container, in that if you have an HA and if it dissociates, not all the HA will dissociate. There will be a part of HA that is dissociated and the part of HA that is undissociated. If we take the ratio of that, we get this. Notice this thing. We have put the dissociated part in the numerator and the undissociated part in the denominator. Okay? It's still bright. Okay. Forget about it. Just concentrate on the slide. Anyways, it's okay if you don't see me also. Yeah, this ratio of the dissociated part to the undissociated part is called as dissociation constant. Okay, now 1 second. Actually, it is called as a constant of an acid. This ratio, dissociation constant, is a negative log of this constant ratio. The ratio is a constant, and if we take a negative log of this Ka, it is called as pKa. That is nothing but an acid dissociation constant. Okay, I'll come into the details. Right now just try to focus on here. Now what we are doing, we have this ratio taken and then we take a log on both the sides. Just a simple mathematics. We have taken log on this side and we have taken the log on this side. No voice? Voice is there? All okay? Okay, do you feel any break in my voice or something? Nothing, right? Okay. When we take the log on both the sides and we try to solve it, what happens here? See, if on right side we are just opening it up, we take log of H plus, and this side we are taking, we are leaving this negative part. This negative part is called as conjugate base. Fine. So what we are doing here is we are just opening the log, and if you see we have a log here. Now what we are doing is, now we very well know this log of Ka is equal to minus, negative log of Ka is equal to pKa. So what we will get, instead of log of Ka, it will be negative pKa. Okay, so this side we get a negative pKa. Okay, and this log of H plus is negative pH. Fine. And here we take the log of this ratio as, we keep it the same way. We'll talk about how that is useful. Okay. Now what we are doing is we are just shifting this pH on this side and pKa on this side. So we get pH is equal to pKa plus log of conjugate base divided by undissociated acid. Now if you see, the measure of conjugate base gives you nothing but the dissociated part itself. It will give you the number of molecules that are dissociated. All of you there, right? Anybody lost the focus till now? Great. So if we try to define this, okay, we have to make this part as 0. If we want to define a pKa, we have to make this log part as 0. How can we make it 0? We very well know that log of 1 is 0. Do you remember log of 1 is always 0? Log of 10 is 1. Log of 100 is 10. You all know these points at least, right? Log of 100 is 2, sorry. Yeah. So the same thing, what we are trying to do is we want this part as 0, so we want this as 1. When can we have this as 1? When our conjugate base is equal to the undissociated part. How can we have that? When, out of the total acid available, half of the acid is dissociated, 50% of the acid is dissociated, we get this as 1. Right? Because conjugate base will equal to the undissociated part. So equal thing divided, we get 1, and then this becomes 0. Okay? pKa is an inherent property of an acid. Yeah, I'll repeat that, Saurabh. Give me a minute. Okay, all of you for repeat. Okay, I'm really troubled by this thing today. Let me repeat it. Okay, we were talking about HA. HA, we put it in a container. Okay, we have how many acid molecules? We have maybe some, imagine we have 8 acid molecules. I'm just taking an example, okay? We'll have a lot of them. Out of the 8 molecules, if 4 are dissociated, that means 4 form H plus, 4 conjugate base, that is 4A minus, and remaining, that is there are 4 that are still undissociated, that is 4HA. Right? Okay. Now if I take the ratio of 4A minus divided by 4, if I want to take the ratio of A minus and HA, I will be getting 4 by 4, that is equal to 1. This scenario is possible when the number of dissociated molecule is equal to the number of remaining undissociated molecule. Right? Why do we want this 1? Because we wanted to define a pH. We are trying to define this pKa in relation to the pH. Yeah, now you understand that statement, Meera. Okay, so this ratio, if we make it 1, okay, then our pH is equal to pKa. So pKa is a pH at which an acid is 50% dissociated. Do you understand that? That is the definition of a pKa for a particular acid. Now what happens is, for example, if you take HCl, HCl will be 50% dissociated at a particular pH. Okay, that particular pH at which HCl is 50% dissociated will be the pKa of HCl, and this pKa will be constant for HCl always. Similarly, for H2SO4 there will be another pKa. Margi, where do you want me to repeat from? I'll just summarize the whole thing once. Okay, all of you be with me. Okay, acid has a property to dissociate into proton and a conjugate base. Everybody understand here? Okay, now if I take a ratio of this dissociated part, now when I use the term, just try to relate the terms to what I am using them for. I am taking the ratio of this dissociated part to the undissociated part. I have the dissociated part in the numerator and undissociated part in the denominator. Okay, I call this ratio as Ka. Okay, now it is said that, that negative log of this Ka, this ratio is pKa. pKa is nothing but acid dissociation constant. Fine? Okay, so to reach that negative log, we are doing some mathematical application. We are applying log to both the sides. We have taken log to one side, so obviously we have to take log to the another side. Okay, so we get log of Ka here, this ratio. Okay, but just now we saw that negative log of Ka is pKa. So log of Ka will be negative pKa. Right, so we get negative pKa here. And log of H plus we get negative pH here. Okay, now we put pH on this side, negative will become positive. We put pK on this side, positive will, negative will become, we are just changing the side, so the sign will change. Okay, now to define the pKa in terms of pH, we are trying to make this side, this part of the equation as 0. If we want to make this part of equation as 0, so that we can define the pKa on the terms of pH, we have to make this ratio as 1, because log of 1 is 0. This ratio of conjugate and undissociated part can become 1 when they are equal. When they are equal, when will be they equal? When the acid is 50% dissociated. When the acid is 50% dissociated, the numerator is equal to the denominator. The dissociated part is equal to the undissociated part. Right? So herein we are getting the definition of pKa, where pKa is equal to pH at which our acid is 50% dissociated. Clear? Okay, we need to move ahead. It is nothing but the same thing written in words. Okay. So this is what we ultimately get. pH is equal to pK when A minus, that is the conjugate base, is equal to the undissociated part, and this is the definition of pK. You don't have to mug up the definition, and if you know the definition you can derive the equation. Okay. pK of a weak acid can be defined as pH when the concentration of acid and its conjugate bases are equal. Okay. Usually a weak acid has a pKa value in approximate range of -2 to 12 in water. A weak acid's pKa is approximately -2 to 12. This is an MCQ. Make a note of this. Acids with pKa value less than -2. A strong acid. Strong acid are the ones that are nearly completely dissociated. They don't exist in that, partly dissociated. They have so much tendency to give off protons that they are terribly dissociated. Okay, they are totally dissociated. And actually, these HCl, H2SO4, they come under the category of strong acid. I was using the term HCl, H2SO4 and all that just so that we can understand what is conjugate base and what is protons. Okay, they are not the weak acid. Usually we come across weak acids in these buffer solution in the system, human body systems. Right? All clear now? Okay. Now, why we are studying about this? Because buffers. What are buffers? Buffers are the ones that have capability to donate the protons and also the capability of taking in the protons, right, as and when needed. Maintain the pH. So how do they maintain the pH? By taking the protons and giving the protons. Right? Buffers, they are most effective when they are used at pH that is equal to their pKa. That is why we have certain buffers which are very active. They are very active at acidic pH. Certain buffers that come into role when we have a basic pH. Okay, we have different buffer systems that and come into play at different pH and different scenarios. Right? Now there is this yellow block. This is just out of the way, to apply the equation. Now if you remember the Henderson-Hasselbalch equation, we have this kind of a relation, right? pKa for a particular acid is always constant. It is a property, an inherent property of the acid. Okay. Now you remember that log of this, how it affects the equation, right? Imagine a scenario if you change the pH by 1. If you just make a unit change in pH, for example, you decrease the pH from 6 to 5, or maybe increase the pH from 5 to 6, whatever. Whenever you make a change in pH, there will be 10 times more change happening in the dissociation, right? Because log of 10 is 1, right? Because pKa is constant. If pH changes by 1, what will happen? We add one more 1 here, plus 1. So obviously this equation has to become 1. When will this become 1? Is when it is log of 10. That means this ratio is 10. So there is 10 times change in the dissociation when there is 1 degree change in pH. That was the question asked in pharmacology. It's given in your Tripathi very beautifully. If you refer it there also, I am very fine. Okay? Yeah. Now tell me, if pH changes by 2, how much will dissociation change by? 100 times. Perfect. Let's come back to amino acid. Why are we talking about all this? Because our amino acids are both acid and base at the same time. Okay, now since they are both acid and base at the same time, they have 2 pKa value, one for the acid and one for the base. Now do you remember that R factor there? That was a side chain. This R side chain is also one more thing, you know, this side chain can have another acid or another base, you never know. Right? If this side chain has another acid or a base, even that will have 1 pK. Okay? Yeah, then 3. But there is a possibility that maybe not. Maybe they are just neutral things. Maybe they are not acids. Okay? We have been talking about zwitterion. Okay, most amino acid at physiologic pH, they are zwitterion, that is they have both positive and negative charge, making the net charge 0, and the pH at which the molecule exists is called as isoelectric pH. We already saw this. A amino acid with only one amine and one carboxylic group, how can pi be calculated? This isoelectric point that we have been talking about, how can it be calculated? It can be calculated by taking the mean of their pKa. If you know the pKa of the amine group, if you know the pKa of the carboxylic group of it, we just take the mean of those pKa and we get an isoelectric point, and that isoelectric point, they will exist in a zwitterion. Isoelectric point is nothing but it will give you a pH at which they will exist in a zwitterion. Okay, that was the basic information about the properties of amino acid and their structure. Let's come across some of the facts related to them. We already saw the smallest amino acid is glycine because it has H in place of the side chain. And one more fact is it is the only amino acid with symmetric or a non-chiral carbon atom. Okay. The second smallest amino acid is alanine. Alanine has CH3 as a side chain. Okay. So the smallest one had H as a side chain. The second smallest has CH3 as a side chain, and that is nothing but alanine. The largest amino acid, here we are talking about the bulkiest one, is tryptophan. Okay, and the longest, the largest chain, be very careful when you talk about the terms. Do you notice the largest amino acid and largest chain amino acid? They are the different things. Now how do I explain it? See, if I talk about the bulkiest one, it may not have a long chain, but whatever it has, it may have a lot of branching and ultimately making a very big molecule attaching at as a side chain, right? But when we talk about the longest chain, it is a chain that is getting longer and longer in one direction. That makes it a longest chain. So tryptophan is the bulkiest one and the longest chain is arginine. Okay, and the second largest is lysine. These are memory based facts, very important MCQs. Proline, as we already saw, was an amino acid because of this cycle which it formed. Remember that, right? And there's one more fact we need to know, that is ninhydrin is the one which is widely used for detecting amino acid. This ninhydrin, it gives a purple product with alpha amino acid and it gives yellow product with amino group of proline and hydroxyproline. Now what happens is this proline and hydroxyproline can be converted to this proline is a amino group, right? This proline can be hydroxylized to hydroxyproline, and whenever it detects an amino group it gives a yellow product, and whenever it detects an alpha group it gives a purple product. Okay, remember this experiment we used to do in biochemistry? I don't know if you all did. Okay, one more fact about proline and hydroxyproline is these are the one which are the basic constituent of a very important molecule, that is collagen. An MCQ which is very commonly asked. The aromatic amino acids which we had been discussing in the chart before, they absorb ultraviolet light. So their presence in protein can be utilized to determine the presence and concentration of protein. So we already know that amino acid, they are building blocks of proteins. If a particular protein has aromatic amino acid, that property, of these aromatic amino acid to absorb the ultraviolet light can be used to measure the concentration of the proteins. And the Sanger was the first one to determine the sequence of a polypeptide. He was the one to determine a complete sequence of insulin, and for that he got Nobel Prize in 1958. The complete sequence of insulin was determined by Sanger. Since we had been talking about aromatic amino acid, let's just look back. We just saw it in the chart before. Histidine, phenylalanine, tyrosine and tryptophan. That chart is very important, guys. You need to mug it up. Then we have something called as modified amino acid. Other than the 20 amino acids which are coded by our DNA, there are certain more amino acids that are generated by modification in the basic 20 amino acid. These modified amino acids, they are formed after the protein is synthesized, and these modifications, since they happen after the protein is synthesized, they are called as post-translational modifications. And let's see what we have in here. There is a question that usually comes: all of the following are post-translationally modified amino acid except. Okay, so in that we need to know what are the post-translational modified amino acid. We have this cysteine. Cysteine is nothing but it is formed by the reaction of two cysteine side chain. This is the sulfur containing, right, to form a disulfide linkage. Okay, and one more point to be noted is only covalent cross linkage found in protein is disulfide linkage. Okay, hydroxyproline is found in the collagen triple helix, as we already discussed, and we also have some phosphotyrosine, phosphoserine and phosphothreonine, which are nothing but the derivative of tyrosine, serine and threonine, and they have a role in signal transduction. We'll talk about signal transduction sometime if we need it. It's nothing much needed as such MCQ point of view. Okay, so we have the examples of modified amino acid, that is cysteine, hydroxyproline, phosphotyrosine, phosphoserine, phosphothreonine. The list is important, guys. Then we have this chart. Probably some of you may have the mnemonic for it. I'm bad at mnemonics, Dr. Bhavna is rather good at mnemonics. Okay, so you have these essential and non-essential amino acids. These are the ones to be remembered again, the memory based stuff. Okay, essential amino acid is an amino acid that cannot be synthesized by the organism. It must be supplied in the diet. But there are certain, these are the ones which are marked in stars. All are essential, but out of this, histidine and tyrosine are essential only in certain cases. They are semi-essential. Perfect. Okay, so if we are supposed to have, so you need to know when, these are totally non-essential ones, and out of these starred ones, they also become semi-essential sometimes. AV, Hill, MPTT. Okay, Hemlata is given, and this thing, it would be great if you could put mnemonics in forum. Maybe it may be helpful for others also, right? Because yeah, here it may be lost. Okay, we took a pretty bit much time for amino acids. If you're good to go, I'll be moving ahead with proteins as of now. And after proteins, probably I'll give you a break before we go to this thing. Hey, how many are they? Let me just count. Yeah, there are 8. True essential. Two are semi-essential. Okay, now when we talk about the proteins, are you guys oriented or lost? Lost in the discussion? Did I miss anybody's question before? Saurabh, I think so it was about pK. You were confused. I mean, that was the point I repeated. All clear? All clear, na? I missed nothing or nobody's questions or anything? Great. Yeah, so next shift to proteins. Proteins, they're classified first. You know, we all know what proteins are. Proteins are nothing but amino acid joined all together to form big ones, that is proteins. Right? So proteins are classified mainly first by their gross structure. In that we have the globular proteins. Globular proteins. Oh, my these things are not showing up actually. Wait a second, let me just have a look. Okay, some of the diagrams are missing, but that's okay. I'll just draw them here. They're not so important. Okay. Okay, if this is a protein, you know, if I take the length of the protein and this dimension of the protein, call it a breadth, shortest and longest dimension, and if I take the ratio, and if this ratio is less than 3, we call them globular protein, and if this ratio is more than 10, we call them filamentous proteins or fibrous proteins. Okay, so these long collagen kind proteins, they are the fibrous proteins. Okay, that's simple. All you have to remember these axial ratios, not over 3 and ratio more than 10. Okay, then other than that we have certain lipoproteins. Lipoproteins have lipids, and glycoproteins, they have carbohydrates attached to the proteins. They are the one complex proteins. And we also have some metalloproteins. These are very important source of MCQs, these metalloproteins. Metalloproteins are the proteins that have these metal ions associated with them. Yeah, matrix metalloproteins. And we have the important one, that is myoglobin, hemoglobins, and cytochromes, and many other proteins. Okay. Let's talk about the structure of the protein. When we talk about the structure of the protein, the first, it is made into various orders. The first order, if we have a lot of amino acid and we just join all those amino acids in chains, what we get is the primary structures. This is the one which is encoded by our DNA sequence. It gives that kind of a structure, right? So if you notice over in the diagram here, it is just a chain. Okay, this long chain, and this long chain is the primary structure. Now what happens is, this long chain then folds into various structural units. Now this long chain could either fold in sheets, or it may fold into helix. That folding into sheet or helix is secondary structure. These geometric ordered unit, do you notice here we have sheet of the long chains, and this is the helix. In sheet what we have, since it is a sheet, we'll have multiple strings, put one after the another, making a sheet, and then we have sheet over one another. That is a typical primary structure of a beta pleated sheet. Okay, then we have the helices, which are called as alpha helices. Fine, that is the second structure of proteins. Now when we talk about the alpha helical structure, these alpha helical structure, they are stabilized by intra-chain hydrogen bond. All you have to remember is this intra-chain. Both alpha and beta pleats, alpha helices and beta pleats, are stabilized by hydrogen bonds. But in helical we have a single chain kind, rolling around each other. So whenever we have a hydrogen bonds in these helical structure, there will be intra-chain hydrogen bonds. But when we talk about the beta sheets are made up of lots of chains. Because of that, we have these interchain hydrogen bonds stabilizing the beta sheets. Fine. So this was the secondary structure. Now we have the alpha or the beta as secondary structure. Now let's arrange this alpha and beta. Now if I arrange small units of this alpha beta, for example, if I make alpha beta alpha, or maybe I make beta beta alpha, okay, or maybe I just make alpha alpha. These small kinds of joining together of the secondary structure, simple combinations of the secondary structure, are called as motifs. Do you notice this? Okay, so that was motifs. Now let's shift to tertiary structures. Now what happens? We have these alpha helices and beta pleats ready. Now if we arrange these alpha helices and beta pleats, okay, to make a large functional units, a three-dimensional assembly, then it makes a tertiary structure. The basic unit of tertiary structure is called as domain. So motifs were just the simple combinations, and once this simple combinations, if they just grow up to make a three-dimensional functional unit, that is a tertiary structure, and the basic unit there is domain. Tertiary structure is a stable structure, and many proteins, they just stay at this tertiary structures, and they just do not progress further. They are just functional at this level. We don't need to go to the quaternary for some proteins. But some of the proteins, some of the large proteins, they go to the quaternary structures, where what happens, there is a typical spatial arrangement. Lots of tertiary structure will come and they'll be arranged in space in typical geometry, giving them a quaternary structure. Is that fine? Our stage is clear? Now we can study the three-dimensional structure of protein by X-ray crystallography or by nuclear magnetic resonance spectroscopy. This is for MCQ. Motifs, yeah, Prasad, motifs are just the simple combinations of these secondary structures. Simple combination. They do not reach to the structural level of tertiary. They are just before the tertiary. Okay, they are just a simple combination, and when the motifs, everything joined together to form a domain, when it's a three-dimensionally oriented and total form structure, then we'll call it tertiary. 3 to 30, I feel, right? Three to 30. Okay. This is just the summarization. Do you want me to repeat, or all clear with that? Shall I go ahead? Fine, right? Okay. Now we'll be talking about two more terms, that is configuration and conformation. Configuration is a geometric relationship between given sets of atoms. For example, we had been talking about this L and D amino acid, right? These L and D amino acid, whenever we need, for example, take a whole chain of protein. If we need to change a configuration of an amino acid inside this whole chain of protein, we have to break the bonds. If we want to convert D to L or something, we'll have to break the bonds. That is the configurational changes in the protein. So conversion of configurational alternatives, it requires breaking of bond. When we say conformation, conformation is just the spatial relationship of every atom to the molecule. The best example I can probably give you is H2O. Remember this H2O molecule, H plus, they exist at a particular angle or something because of the charges and all those things. That gives it a conformational arrangement, and that could be changed without breaking the bond. Okay, and many a times what happens, if we have a single bond, single bond joining two atoms, there could be rotation of these atoms around the single bonds without their breaking of the bond. In such cases, these kind of changes are called as conformation changes, because it doesn't need breaking of the bonds. It's just the changing of the arrangement of the atoms inside the molecules. Fine? So whenever we have a denaturation of a protein taking place, what happens, there is a loss of native conformation resulting in a random coil. So what is lost is just the conformation lost. But what happens, this conformation of the protein is important for its function. If the conformation is lost, it becomes just randomly oriented and the proteins are not able to function. And such proteins, such denaturation is caused because of pH and detergents and all those things, boiling. Okay, yeah. So that was about the configuration and conformation. We'll be talking about three of the important proteins. Out of this, the first one is chaperons, and these chaperons, they are the proteins which are important in the way that they assist the non-covalent folding or unfolding. What does that mean? Do you remember the folding of the secondary chain when we were talking about the secondary chain, wherein we were talking about hydrogen bond forming the beta sheets and all those things? Right? If you remember, in those hydrogen bonds, even they are responsible for folding of the chain. But they go and sit in the chain. They are the one actually, when they fold the chain, they become the part of the chain. But these chaperons, they do not get incorporated in the proteins which they are folding. They only promote its folding or unfolding, or prevent or promote depending on the scenario. They do not themselves go inside the protein which they are affecting. Something like an enzyme, remember? Yeah. So, but one more point about these chaperons is, they do not come into role when the protein is functioning normally. They usually come into the scenario when protein is stressed. For example, when we talk about the proteins, nearly everything in the body is protein. Some enzymes and many things, whatever function, they are just proteins. And whenever there is some kind of a stress, maybe it's a bacterial stress, microbiological stress, or maybe it's a heat stress, or maybe it's it could be thermal, anything, any kind of stress will make these chaperons come into the picture, and they will prevent the proteins for getting damaged because of such kind of shock by managing their folding and unfolding. That is why chaperons come into the scene in stress, and that many a times they were called as heat shock proteins. Okay, now we very well know that there is a tendency of the proteins to become disorganized or to lose their configuration in presence of heat. Okay, so whenever such thing would be happening, these chaperons will come into the picture, and they will make sure that the folding is proper, and there is no haphazard folding of the proteins, and that is why these were called as heat shock proteins. Many of these proteins are also expressed by bacteria. Many bacteria, they have these heat shock proteins to protect their proteins from getting damaged. You understand what is chaperons? Kind of. Okay. Then we have this hemoglobin. We all know what hemoglobin is. Let's just see its structure in the perspective of protein. Hemoglobin is an iron containing oxygen transport metalloprotein because it has metals and proteins in the RBCs. What it has? It has proteins that are called as globin. Okay, it has 4 globins, 2 alpha and 2 beta. Now at this point, don't confuse this alpha and beta to be alpha sheets or beta sheets. Alpha and beta do not apply to beta sheets or alpha sheets here. They are just the 2 globins are alpha globins and 2 are beta globins based on their total structure, total chains. Yeah, alpha chains and beta chains of globins. Right? Other than that, they have this heme group sitting inside the globin. So they have 4 globin chains, 2 alpha and 2 beta, and they have 4 heme groups sitting inside these globin chains. Now if you see these globin chains in a bit more magnification, what do you see? That they are mostly made up of alpha helices. Now I am going inside, okay, I am talking about the secondary structure inside these globin chains. The secondary protein structure inside the globin chains, be it alpha chain or beta chain, both chains have alpha helices inside them. Okay? Don't get confused, okay? Even beta globins, they have alpha helices inside them. One more thing that we need to know at the heme part. Heme part is the one that is, this part, this green colored part that holds the metal. We'll talk about that in next slides. This heme part is synthesized in mitochondria and the cytosol of immature RBCs, while the globin part, it is synthesized by ribosomes. We all very well know that protein is synthesized in ribosomes. Okay, now let's see what is heme. Heme, we have both the spellings, H-E-M-E and H-A-E-M. It is a prosthetic group that consists of iron atom contained in hydrophobic center of the large heterocyclic organic ring called porphyrin. So this ring which you see here, this blue colored ring, is nothing but porphyrin ring. In porphyrin ring, in the center you have a hydrophobic pocket. There is an MCQ on that. A hydrophobic pocket of the porphyrin ring contains iron. Anybody has come across that? You will come across that in biochemistry somewhere. Okay? So this hydrophobic pocket contains iron. Now what is this ring made up of? This porphyrin ring, it is made up of pyrrole subunits. Right? So we have these 4 pyrrole subunits joined together by methane bridge. So these methane bridges, they are connecting the alpha carbon atom of every pyrrole ring. So we have 4 pyrrole. Do you notice over here in this diagram, you see the methane bridge very well, right? And they form a ring, and in between there will be a hydrophobic pocket that can hold a metal. All clear till now? Okay. So now if we talk about in a sequence, we have these pyrrole. This is bad actually, this thing. Okay. Pyrrole, 4 pyrrole, they join together to form protoporphyrin, a porphyrin you can say. Okay. This porphyrin has a pocket in the center that contains iron. So these porphyrin with an iron in between totally makes a heme group. Right? This heme group sit inside the globin chain. Such 4 globin chains with each heme group sitting inside them come together to form hemoglobin. Right? Have the structure of hemoglobin clear? Because structure of hemoglobin is very important. Very important. Okay. Now, remember when we were talking about breaking of RBCs? RBCs have these hemoglobin inside them. When the hemoglobin breaks up, globin is separated and heme is separated. You all very well know what heme contains now. It contains porphyrin and iron. Iron is again taken out. Iron goes back for making another RBCs. What happens to the remaining thing? Remaining thing was nothing but these 4 pyrrole subunits. This cyclic ring is opened up, and what we get is these pyrrole subunit. They form a straight chain because of opening up, and this straight chain, this pyrrole ring is opened up, and this pyrroles, it is converted to biliverdin. Okay? Biliverdin is converted to bilirubin. Bilirubin goes to liver. In liver it is conjugated with glucuronic acid. From there, this conjugated bilirubin is released by bile into the duodenum. In duodenum, because you know whatever liver forms, it goes into the gallbladder concentrated, goes back into the duodenum. In duodenum again, when it goes down there, again this conjugated bilirubin is deconjugated. Yeah, deconjugated, and then we have the stercobilinogen and urobilinogen form that gives color to the stools and urine. All clear? You know, hemoglobin part and all those things, it would have come across in physiology. I remember taking that myself rather, and all those things. So it is very important. It comes in physiology, it comes in pathology, it comes in biochemistry. Right? So bilirubin is a breakdown product of heme, consists of an open chain of 4 pyrrole ring, that is tetrapyrrole. Great. Now when we talk about the myoglobin, myoglobin is a compound very similar to the hemoglobin. Only difference is it has a single chain globular protein, and it contains a heme, the same heme, in the center of the protein. So there is only one chain there, not 4 chains. Right? CEL. This point we have here, it has a molecular weight of 16,000 Dalton, that is 16 kilodalton. This is comparison of hemoglobin to myoglobin. Where do you find myoglobin? Yeah, it's in the muscle. Yeah. Then we have one more structure, that is cytochromes. Cytochromes are again membrane bound hemoprotein that contain heme group and carry. You know, these are again, they have a similar structure, but if you remember, in the mitochondrial membrane there is an electron chain that operates, and this electron chain operation is carried about by these cytochromes. We'll be talking about cytochromes and electron chain later when we come across the metabolism part of it. Okay? Here we are finding about the basics of the proteins, and with this next we'll be starting with carbohydrates in maybe another 5 minutes break. CHO, that is CH double bond O is your aldehyde group, and this C double bond O, when it exists in the center, it is ketone group. Okay, we'll see that in the next slide. It will be in the diagrams, it will be more easy. Other than that, it will also have multiple hydroxyl group. We very well know what are the functions of carbohydrate. Stores energy, it is a part of DNA and RNA as ribose and deoxyribose sugars, and it also forms the cell wall structure of the bacteria and plants. It is linked to proteins and lipids, and then these lipoproteins, glycoproteins, they have various functions, and it has a major role in cell-to-cell recognition also. So with these basic things, we go ahead and let's see what the general formula of carbohydrate is. CH2O, n number of times. N tells you the number of the carbon. How this structure would be expanded, you will have an idea as and when we go ahead with the slide. So, but this is a very direct MCQ that has been asked in exams. Okay, so it is CH2O n number of times. Now, basically these carbohydrates, they are derived from the parent compound, which is an alcohol. Now, from an alcohol, when we oxidize it, we can get an aldehyde or a ketone based on the structural changes. Now, when we write in just this kind of a formula which we had been talking about before, it doesn't actually give us an idea whether we have an aldehyde or ketone. Aldehyde and ketone depends upon the position of the double bond. Okay, how that happens, let's see over here. The example which we are taking is glycerol. Glycerol is 3 carbon structure, and when we just oxidize it, we can get an aldehyde or an ketone. Aldehyde and ketone both have a group, that is carbonyl group. Carbonyl group is nothing but C double bond O. Okay, when this carbonyl group, is in this kind of a thing, wherein this carbonyl group has on one side, it has a side chain, and on the other side it has hydrogen, then this makes an aldehyde. But when this carbonyl group has side chains, that is R group, on both the 2 remaining bonds, then it is a ketone. Okay, are you getting me on this? How this happens, when we'll see in other groups, will come. One very important thing which you have to always remember, because we are dealing with carbon. Carbon has 4 valencies. If you remember that, it can form 4 bonds. So it is forming here double bond with oxygen and single bonds with this side chains. Okay, so now let's see how glycerol is converted to aldehydes and ketones. So this is the glycerol, that is an alcohol. Okay, that has 3 carbons, and its valency is satisfied by 3 OH and remaining by H. So when it is oxidized, it gets 1. How it is oxidized? By removal of 1 hydrogen. See, oxidation is defined as addition of oxygen or removal of hydrogen. Hopefully you know the definition of oxidation. So since we are removing a hydrogen, now this hydrogen is removed, so 1 valency of the carbon which is unsatisfied, it will come back and form, and 1 valency of oxygen is unsatisfied. But both oxygen and carbon have a tendency to form double bond also, so they form a double bond here. Okay, because of the formation of double bond, we get a carbonyl group. All of you getting these points? This carbonyl group, when it is formed with this terminal carbon, this terminal carbon will have the carbonyl group, and one side it will have H, and one second, this part will be nothing but R, so it will be an aldehyde. That makes a D-glyceraldehyde. D and all we'll talk about later. Okay, right now what you have to understand is it makes it glyceraldehyde. Now when we talk about COO here, if it forms with the center one, if you notice, if it is forming with the center one, This was the central carbon, and if the central carbon loses its H, the double bond will form in the center. Now this carbonyl group which is in the center will have one R here and one R here. That makes it a ketone, right? So how you can identify an aldehyde and ketone? By a very simple thing. If your carbonyl group is present with a terminal carbon atom, it will be an aldehyde. If it is present in non-terminal carbon atom, it will be a ketone, right? So this is how we now, but if you just notice, you know, if you can make that formula, it will be C3H, how many H? 1, 2, 3, 4, 5, 6, H6 and O3. Can you make the formula for ketone? Even ketone will make a same formula, right? The number of carbon, hydrogen and oxygen will be same in aldehyde and ketone. All that is different is the position of the double bond between carbon and this thing. Okay. Now, based on the hydrolysis, this part of the classification is simple, so I'll just brush through it fast. We all know what is monosaccharides. They are the one which are hydrolyzed totally and cannot be further hydrolyzed. And in that, based on the number of the carbon atom, we have trioses, tetroses, pentoses, hexoses, heptoses. Hopefully you all know all these points, right? And then they can have, you know, all trioses, tetroses, everything can have aldose form, that is aldehyde form, and ketone form, as we already saw. Then we have disaccharides. They are nothing but two monosaccharide unit coming together and forming. In that we have maltose, sucrose, etc. We have oligosaccharides. Oligosaccharides are the product of 2 to 10. More than 2 to 10. 2 to 10 are oligosaccharides. Oligosaccharides which have two things are called as disaccharides. 2 monosaccharides are disaccharides. 2 to 10 is oligosaccharides, and if they have lot of them, more than 10, then they are polysaccharides. Okay. Now these monosaccharides, we'll be talking about monosaccharides first. Probably we'll be able to finish monosaccharides today. Let's see if we can go ahead a little more. These monosaccharides, they have these aldose form and ketose form, as we already saw with glyceraldehyde. Okay. So in that we have this example of glyceraldehyde. Aldose is glyceraldehyde, and ketose it is called as dihydroxyacetone. The names are important. Okay. Ketone form of glyceraldehyde is dihydroxyacetone. Now if you notice this carbon atom which is circled over here, okay, let me mark what is attached to this carbon atom. The aldehyde group H, OH. So do you notice it is a chiral carbon atom? It is an asymmetric carbon atom? Yes, it is a chiral carbon atom. But if you notice here, what it has? Here it has O, but notice these two, they are same. Yeah, so it is not a chiral. So if we have a chiral, we get the isomers. Remember those isomers which we had been talking about in amino acids? It's similar over here. So we will get two isomers in glyceraldehyde because of that exchange. But do we get any isomers in dihydroxyacetone? No, we do not get, because it doesn't have a chiral carbon atom. Okay. This we are doing with the simplest sugar, that is glyceraldehyde. Now when we go ahead with a little complex, or whenever we increase the number of the carbon atom, we'll see how the things change. Okay, the aldose form has a chiral carbon which is encircled here, so it can have two isomers, the D and L isomers, but ketones have no chiral, it doesn't have an isomer. Okay, Now the isomers of the monosaccharide, they depend on the number of carbon. Before I go to this slide, I'll explain you a little more on the whiteboard. Let me make 4 carbon, a tetrose. Okay. Okay, now if I want to make, I have 2 carbon in the center. One is this one, and the second one I'll mark in blue. One is this one. Okay. Now let me just mark for the blue one. Let me mark the side chains for the blue one. This is one side chain for the blue one. Okay, actually I'm supposed to make OH and H here also. I'm sorry. This is one side chain for this carbon atom, blue wala. This is one side chain. This is one side chain, and this is one side chain. Are you getting me on this? What I'm trying to say is the chirality of this carbon. Now let's see the chirality of the other one, which I marked the circled red. Let me make its chirality in green color. Okay, this is one side chain of that thing. Yeah, this is one, this is one, this is one. Alright, there are 2 chiral. Okay, now let's go with this thing. Is this chiral? The one which I am marking in purple, is this chiral? No, because it has 2 H. If you notice, it has 2 H, it is not chiral. So total in the one with 4 carbon, we have 2 chiral carbons. Right? Okay. So the isomers of the monosaccharide, it depends on the number of these chiral carbons. Okay, so how do we calculate it? There is a formula that says 2 raised to the power C, where C is nothing but the number of chiral carbons. Okay, it will give you the number of isomers it can have. For example, when I say aldotriose was aldose, that is nothing but the glyceraldehyde. If you remember, how many chiral carbon it had? 1. So if I make it 2 raised to the power 1, what I get is 2. So it had 2 isomers. But remember the ketone form of glyceraldehyde, how many chiral carbon it had? Nothing. So 2 raised to the power 0, it gives me 1. Yeah, but in the last example, that was about the tetrose. Okay. So this is the tetrose, aldotetrose. In aldotetrose, we had 2 chiral carbons. So 2 raised to the power 2 gives us 4. Right? But if we make the ketone form of the same thing, what will happen is ketone may one double bond will come somewhere in the second carbon. Because of that, we'll only get 1 chiral carbon. You can actually make the structure yourself and find it out. If you have understood the structure, you'll be able to, make it actually. It's simple. Since there would be only 1 chiral, 2 raised to the power 1 will give you 2. Okay, so number of chiral carbon determines how many numbers of isomers, the DL and isomers, it may have. Are you all clear with this? Great. Okay, so here we have some of the important sugar. We have some trioses. In trioses, we have glycerose, that is glyceraldehyde. Then we have dihydroxyacetone. In tetroses, we have erythrose and erythrulose. Pentoses, we have ribose sugar and ribulose. hexoses we have glucose and fructose. Fructose is nothing but a ketone form of glucose. They have the same structural formula, but because of the presence of double bond at the different point, it is the fructose and this is the glucose. And they have different properties, okay? Because of being ketone and aldehyde, they have totally different properties. Some of the important monosaccharides, we have D-glyceraldehyde, as we already saw. It is the simplest sugar, the enaldotriose. D-glucose is the most important in diet. D-fructose is the sweetest of all sugars. D-galactose, it's a part of the milk. D-ribose is the one that is used in RNA. And deoxyribose is a ribose that has lost oxygen at the second carbon. Now ribose is nothing but 5 carbon atoms sugar. Okay, if it tends to lose one oxygen, it will become deoxyribose. And at what position it is losing the oxygen? At the second carbon. An important MCQ here. Now let's see various forms of isomerism. You know, we had been talking about D and L isomerism. Now as far as DL and isomerism of amino acids, a similar concept apply here, but there is a little more restriction here. Let's see what those restrictions are. Now when we talk about D and L isomerism, here what we consider is, we consider a carbon atom just adjacent to the terminal primary alcohol carbon. Okay, so this is your last alcohol carbon, and this carbon, you know, this carbon we'll consider when we consider D and L isomerism, just the carbon which is next to the last terminal primary alcohol. Okay, when in this second last carbon, if we have OH on the right, it is D sugar. When the OH is on the left, it is L sugar. Getting me on this fact? Yeah, see, OH could be right and left at so many places. If you notice in this particular glucose and fructose, there are so many carbon and H and OH. See, we are not bothered about the position of these OH and H. We are only bothered about the position of the fifth carbon in glucose. Fifth carbon is the glucose just adjacent to the terminal alcohol carbon. Terminal alcohol carbon is this one, and this is the second just adjacent to that. So D and L isomerism depends on only this particular carbon which is adjacent to the terminal alcohol carbon. Clear? Now, okay, now if you notice this D, okay, now we have other, you know, this was the first, this was the fifth one. Then you have these fourth, third and second. Do you notice this? Let me number them. This was your fifth carbon which determined your D and L isomerism. But we also have this fourth, third and second, right, which can turn around themselves, which are the, you know, wherein we have the chiral carbons and they can turn around themselves. So we can also have isomers with these, but they are not called D and L. Okay, we'll talk about them in the upcoming slides. The chiral carbon determines the D form, OH on the right and L form on the left. The biologic form usually is the D form. That is why if you remember in that chart wherein I was talking about this, the simplest sugar, the sweetest sugar, we were all having D sugars in there, right? Yeah. The other forms, that I was talking about, the second, third and fourth chiral carbon atom, whenever we have changes in them, those are called as epimers. The epimer, isomer that differs as a result of variation in configuration of OH and H carbon at second, third and fourth, they are called as epimers. So at fifth carbon what you get is, in glucose we are talking about, in glucose at the fifth carbon we are getting is D and L isomerism, and at second, third and fourth whatever isomerism we get is epimers. Okay, now for example, if you notice these three things, here this is our fifth carbon, right? In the fifth carbon we have OH on the right side. In all three we have OH on the right side. All three are D forms. Okay, now in the D forms, this is the D glucose. Okay, in D glucose, this is the fourth carbon. In the fourth carbon, if I change H and OH, rest everything remaining the same, what I get is galactose, which is an epimer of glucose at fourth carbon atom. Getting me? So D-galactose is an epimer of glucose at fourth carbon atom. Both are D. Both are D because at fifth carbon both have OH on the right side. Now if you notice here the second carbon, as compared to glucose, mannose ka second carbon OH and H is interchanged. So mannose is an epimer of glucose at second carbon atom. So it is D-mannose. Similarly for L-glucose we will have L-galactose, L-mannose, etc. Right? So that is why you get so many epimers and isomers. You know, when we talk about the glyceraldehyde, see here. No, don't get confused. We are just this is the level of glyceraldehyde. Okay, this is the level of four carbon, this thing. Don't get so much confused. But hopefully you understand what is the difference between. You don't have to, you know, actually when you start drawing it, you know, it's very easy. You can yourself draw everything and get back the answer if you get such a certain complicated thing. But usually the questions are very direct. What are epimers? What is the relation of galactose to glucose? Now you all know that it is not an D or L isomerism that is different. You know that it is epimerism. All you have to understand what is epimerism and what is isomerism, and you have to know a few facts about them. For example, the relation of mannose and glucose, the relation of galactose and glucose, and that way. That's it. Not that difficult, right? Okay, then we have optical activity. Optical activity, I had been talking about optical activity. Remember we had been talking about optical activity being dextro and levo. But I told that, you know, when the discovery was made, they were the D and L, but later they found that, you know, the D and L did not actually fit, you know, the structure, and optical activity did not actually fit in. So what they did was, although the dextro-rotatory and levo-rotatory term was itself maintained, but the two isomerism, the structural isomerism and optical isomerism, were separated out. So that is why we get another kind of isomerism in glucose, that is the optical activity. The presence of this activity is also there because of the presence of asymmetric carbon atom. But when a beam of plain polarized light passes through a solution of an optical isomer, I mean there are two solutions. The two solutions are isomers of each other. They are optical isomers of each other. One of them will rotate to the right and one of them rotate the light to the left. Okay, the one that rotates to the right is designated by plus sign, and the one that rotates to the left is designated by the negative sign. and that has nothing to do with D and L isomerism of the glucose. Because D and L isomerism of the glucose was based on the 5th carbon atom position of OH and H. Okay. The direction of the rotation is independent of the stereochemistry of the sugar, so it may be designated at, see, D may be minus, D may be plus, L may be minus, L may be plus, depending on its effect on the optical light. Okay, so this is the third type of isomerism we saw. What? Prasad, ma'am, and 3D, D or L form will be similar or an optical phenomenon? I'm not getting you, Prasad, actually. See, D and D and L form is a separate isomerism in total, and optical isomerism is a separate isomerism in total. Optical isomerism is independent of that D and L isomerism. There are various ways. That's what I'm trying to tell, it was during discovery of the optical activity of certain chemicals, it was found out that way. Later what happened, as we progressed further, it was found that no, this D and L isomerism and this stereochemistry that we defined of being left and right are actually not so interdependent. So they separated those isomerism. The term D and L still got attached to the stereochemistry part, and optical activity was then denoted by plus and minus. So this is a total different isomerism. So one isomerism was D and L. The second isomerism we were talking about was epimerism, that was at 2nd, 3rd and 4th level in glucose. Yeah, a little confusing, Margi. You have to actually separate out. I know it's a little confusing. I agree with you all. But I just want you to separate out, because we always talk D and L is, here in glucose, in sugars, at least you have to separate it out. In some compounds, the optical activity may be dependent on that direct D and L. But right now, when we talk about the carbohydrates, they are just totally independent. Right? Yeah, now we'll be forming the ring structure. Okay, are you all with me? Yeah, because I want to complete the isomerism, then we'll stop. Okay, I think the biochemistry is gonna be a bit long again. We never used to cover biochemistry as such. What we used to do is we used to go retrospectively, considering the fact that these are the basics, and people later we realized that the basics of the chemistry were really in a bad shape, so we started like having descriptive classes for biochemistry also. So we have lots to cover then since we started. Okay, let's talk about the pyranose and furanose ring structure. In pyranose and furanose, what has happened? There are two ring structure. If you remember glucose, if you just remember your books, you may have seen glucose drawn as a straight line, which we had been seeing till now, and you would have noticed the glucose written in a ring format. Okay, how does that happen? How does that happen? We very well know that glucose has aldehyde at one side. Do you remember at the other end glucose has alcohol? At one corner it has aldehyde and one corner it has alcohol. There is a tendency of alcohol to combine with, react with aldehyde to form hemiacetal. Okay, so normally an alcohol can interact with aldehyde to form hemiacetal. So similar thing can happen in glucose itself. Glucose or any molecule, you know, when I talk, I have a tendency to talk, say glucose. Okay, please broaden your receptiveness to apply the same thing with other molecules also. Hopefully I don't restrict you there. Okay, so whenever we have a carbohydrate, we have one side as aldehyde and other side as alcohol, and there is a tendency that this alcohol goes and reacts with that aldehyde, forming a hemiacetal, and because of which there would be a formation of ring structure. Do you see this? Okay, what has actually happened is, this was the 6th carbon, this is the 6th. Do you notice this is the 6th? And this is the 5th. Okay, so this 6th remains at one end, and there is these 5 carbons coming together, and the 6th end is formed by this oxygen. Okay, so it's a 6-cornered ring, it's a hexagon formed when we talk about the glucose. Now come back to fructose. Do you notice in fructose, C double bond O is not at the corner? It's not at the corner, it's here. Now when fructose tries to form a ring, where it will form interacting at the 2nd carbon, because double bond is there. Because of this, what happens, it forms a pentagon. And this is the 1st carbon lying here, and this is the 6th carbon lying here. Clear, all of you? Are you able to see this? You know what happens, we have a tendency that we don't like to see this CH, OH, OH, CH, OH, 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 CH. So we just don't see them, we don't look at them. Even when we read biochemistry, we don't look at those diagrams. Many of you, I feel, you know, many of you just read the text, somehow get something out of the text, but oh my god, I don't want to look at those diagrams. But they're not so bad, you know, if you just look at them, yeah, if you just look at them with a little, you know, affection, maybe they'll, they'll be more simpler. Okay. Oh, good, Kirti. Then it wouldn't be so bad for you. Okay. So you all understand now how the 6 ring and 5 rings form? Yeah, I'm finishing, you know, I have to make your isomers go in one class. I can't split it in 2 classes, otherwise it will be, I have to restart with isomers again. Okay, I'll just have a to end it. Okay, now after the cyclization, after the cycle is formed, I am very bad at pronouncing that term, cyclization based on the position of OH. Now we can have 2 more isomers. These are called as anomers. This is based on the position of OH. See this C1, first carbon, yeah, first carbon of glucose just adjacent to the oxygen. Okay, if you notice on one side of oxygen, you have this 6th carbon, that is pura CH2OH. Okay, so we are not considering the one that has that CH2OH. We are considering the carbon adjacent to the oxygen that has H and OH attached to it. Okay. So in this case, if the OH is positioned down, or it is also called as trans to CH2OH, it is always in relation to this. Do you notice CH2OH here is on the top? If your OH is away from that CH2OH, then it is alpha form. And if OH and CH2OH are on the same side, it is beta form. And these anomers, these are called as anomers, alpha and beta anomers, they can be seen only when we have the cyclic ring. That is an MCQ, very direct MCQ. When do we see anomers? After cyclization. Right? Simple one. Or if you know it, you will answer it. Okay? This 5-ring structure is, is the furan structure. It is called as furan structure, and 6th ring structure is called as a pyran structure. This is direct MCQs again. Furan, pyran. Okay. Then we have these conformation. Remember, we had been talking about conformation in proteins. It's the orientation in the space. I had been talking about the same as H2O. Do you see this ring? It's lying flat on the surface. Okay, I actually made that whenever I draw, people don't understand it, so I thought I'll bring a diagram there. Now this is my replication of this ring. Okay, when I see it from the top. Okay, when I see it little from the side, I'll see something like this. I mean the 6th thing, little from the side if I notice. Now if I just happen to pick this corner a little up from the ground, this is the ground, this is all attached to the ground. Okay, if I lift this corner away from the ground, giving an angle at this point, getting me? This is a flat thing. If I lift one corner, okay, and lift this corner also upwards, giving it an angle here, when I look it from the side, it will look like a boat. That is a boat conformation. Okay. Now rather than bending it upwards, one corner if I bend it down, it becomes chair conformation. Easy, right? Now let's see the facts about it. The ring structure can have conformation of boat or a chair. Chair is more favored conformation because there are less steric hindrances. See, why do we have these conformation? We have these conformation because we have so many molecules of different charges over there. They repel, they attract and all those things. That is why we have various conformation, and out of this, the chair is a more favored conformation. We'll not go into the details how charges go on. Actually, if you remember in the chemistry when we used to read organic chemistry, do you remember drawing those flower-like things? I used to love drawing them in my chemistry in 12th. They were nothing but that is where we read the conformation. Some flowers were broad because of some, charges and all those things. Right? Yeah. Electron clubs. Yeah, kind of. So those flower things are nothing but conformation, and that is how the so I am not going into those details. All you have to know is that the chair conformation is more stable for this thing. See, if you notice the chair form here, this is just the ring thing, bent down, and one part of the ring is bent up. And this is the boat configuration, both the thing are bent up. Okay, so these are the two conformations. Enough for today. Do you all remember the isomers now? The first was D and L isomerism. The second one was epimers at second, third and fourth when we talk about the glucose. The other C's, other chiral carbons, and then we had been talking about the optical isomerism, plus and minus. Then we had been talking about the anomerism. Anomerism, that was present only when we had the ring structure. Yeah. Then we saw about the conformational changes in those ring structure. There is one more point over here. You can just read it out in the notes also. Furans, they adopt a typical envelope configuration. What happens is this is 5 ring structure of the furans, right? So this part, I'll just draw it sidewise. This part, it folds up at this point. So it's something like an envelope folding, like that way furans adapt that envelope kind of a structure. Right? All clear till now? Thanks, Soumya. Great. Sure, Anindya, I'll look into your question. Yeah, I'll look into the questions. Okay, so again, I just assume that in biochemistry we may land up having at least one extra class, at least one. Next class we'll have on Wednesday.